Okay, good. All right. Now, those questions should look familiar. People didn't see any of them on your quiz last night? No? You didn't see, you don't, those questions didn't look familiar at all? Some of them? They all do? Oh, all right, okay. Well, I'll make sure your test has questions that don't look alike at all. All right, okay. We'll make it happen. All right. All right. So we left off, we'll pull up some videos in a little bit, right? But we left off in here with some stuff on metabolism. So first off, we're going to head off into food micro now, a little bit of a food micro unit. But we tie it in with metabolism because we're going to talk about fermentation today. Now, back in chapter 6 on metabolism, definition of fermentation is what? What's a classic sort of definition? Remember, we had glycolysis, right? And you took the glucose molecule, you split it into half to get 2-pyruvate. And then at the end of that, end of glycolysis, if you were an obligate fermenter like streptococcus, your only choice was to ferment. Okay? And fermentation, by definition, was the use of an organic compound as the final or terminal electron acceptor. So the example from last class with streptococcus was at the end of glycolysis, you get a couple of ATP, you get a couple of jacked up niacins, NADH, all right, and then you end up with these 2-pyruvate. But for glycolysis to continue, those niacins that have some hydride ion, right, hydrogen on them, and some electrons, for them, they have to recycle. To do that, they have to ditch those electrons. And remember, if you're an obligate fermenter, all you've got is glycolysis and fermentation. So in the case of strep, they take one of those pyruvates, which is an organic compound. The NAD, the niacin, ditches the electrons on them. It becomes NAD plus, goes back to the top of glycolysis, and they can break down the next molecule of glucose, right? That's the classic definition of fermentation, right? Again, right? An organic compound is the terminal electron acceptor. So just right, how about everybody got this right in the quiz? How about anaerobic respiration? What's the terminal electron acceptor for anaerobic respiration? Is it Miss O'Neill? Is that right? No. Last name? Phillips. How about for anaerobic respiration? Don't remember? How about aerobic respiration? What's the terminal electron acceptor? Oxygen, right? And for anaerobes, it's what? Sulfur, nitrogen, something else, okay? Inorganic, okay? All right. Okay, so what are we talking about fermentation today? Because when we talk about food micro, we're going to talk about fermented foods. Anybody here ever eat yogurt? Right? What's yogurt? Man, it's milk products that are fermented, right? And of course, depending how you ferment it, how long, yogurt can be, like some of that Yoplait stuff with 10 tons of sugar, pretty sweet. Or it can be some very unsweetened sort of sour types, right? Where there's much more lactic acid produced, okay? All right, so about fermentation. Let's see over here. Okay, all right. So a couple of terms here. So as you're reading through, right? So this is chapter 30, right? In the new text, number 9th edition was 31 for a long time. All right, so we're going to talk today. We're going to talk a little bit today. We're going to just go through a few definitions, get a little bit on food spoilage, all right? And then we're going to spend a little bit of time on FBI, okay? Foodborne illness, all right? And that category, FBI, breaks into 2 subcategories: foodborne intoxication and foodborne infections. So you got 3 FBIs, right? The first is foodborne illness, category-wise. Then you can have foodborne intoxications, and then foodborne infections. Okay. Who's ever had food poisoning in here? Right? So what happened? Did you get to vomit? Good, right? Okay. Did you feel nauseated? Yeah. Of course, right? Did you get any diarrhea? No. What's that? Right. So what type of food did you eat when you got sick? How long after you ate that food?
Like a couple of hours.
Perfect. How about you, Ms. Burnett? Right? So what is that? Is that a foodborne? It's foodborne illness, obviously, right? But it's key, right? When you get to the emergency room, start working there, is to ask the patients what's the cutoff on the time. Both of these young ladies were sick within a couple hours. So is that an intoxication or an infection? Right? If you go out to the bars tonight and down a fifth of scotch, what's going to happen to you? You might have a good time. You may not. Depends if you remember it, right? But you're definitely going to get what? Intoxicated, right? It's going to hit you quick. So foodborne intoxications, you ingest a preformed exotoxin and you get sick like these two people did, which is classic, within a couple of hours. And you get nausea and vomiting, not so much diarrhea. The cutoff is about 6 hours. Sometimes you can go as far out as 8, but if you work in the ER, one of the first things the patient come in and tell you that they've been food poisoned is the timing. Less than 6 hours, you know it's got to be a foodborne intoxication. For foodborne infections to happen, the second part of it, usually they ingested something and it's usually about 18 hours later. And the difference is they ingested the bacteria. So again, when they ingest enough of the bacteria, those bacteria then have time to colonize part of your small intestine, multiply, then produce a toxin. On the first one, foodborne intoxication, you ingested the preformed exotoxin. Okay, now I hate to tell both of you this, depending on what food you were eating. Okay, what was it? Do you remember? What?
It was small Chinese chicken.
Okay,
Yeah.
all right. Any rice in it?
No, it was just
Chicken. All right. And for you, Miss Prenda? Okay. Right. So you right. So most likely, what happened is this: is one of those Staph aureus in someone's nose, right? Fell into that chicken, okay, or someone did drilling, then made your chicken, right? And what happens then, maybe is that again the bug multiplied, okay, in the chicken. Now that bug then secretes, that bug being Staph aureus secretes a exotoxin. And the problem with Staph aureus foodborne intoxication is that exotoxin is heat stable. You got to remember that because most proteins, what happens when you crank up the heat? They denature, right? Take an egg, put it on a hot burner. What happens, man? That albumin in the egg goes from clear to what? White, right? It denatures it. But with Staph aureus, the exotoxin they make, okay, and what's the exotoxin against just a protein? It's heat stable, meaning that you can still heat that stuff. So my guess is whatever the chicken you both ate was probably either been cooked and reused, right? Or again, because it needs time for the bug. The staph, right? It wasn't the staph that got you sick, it was their exotoxin, okay? Now, category wise, whenever you have a toxin that gives you NVD, nausea, vomiting, diarrhea, they call it an enterotoxin. Entero meaning your intestine, okay? And really, Staph aureus has historically been the most common foodborne intoxication in this country. But that's changing now, too, because we eat a lot more rice in this country. And with rice, there's a bug called Bacillus cereus. So we all know about bacillus. What do they make? What's a unique feature for bacillus? Spores, right? So with Bacillus cereus, that's why I asked both of you about rice. So Bacillus cereus, what can happen is they heat up the rice early in the morning. And if you ever had like rice, right, you have to take it, wash off the talc, you put it in your steamer, you cook it up. But if you let it sit there all day, as the temperature comes down, the spores on those grains of rice, right, germinate. And if it sits there 12, 16 hours, then those bacteria, Bacillus, right, cereus is the bug, cranks out a exotoxin. Then you eat it, right? And then you ingest that preformed exotoxin. And, right, since we have 2 volunteers for next week, I'll bring in some infected rice, okay? So we'll eat it here beginning of class, and I'll make sure it's so toxic that before class is out, you're both sharing your love with this, okay? Right? Because the same thing's going to happen. You're going to get an enterotoxin that's going to be just like Staph aureus, nausea and vomiting, right? Now, okay, so foodborne illness. So infection, of course, means the bugs, you have to eat enough of them to colonize your small intestine. Because most of these bugs that cause these foodborne intoxications and infections, they don't belong in your GI tract, right? Where do they belong? And they'll maybe your colon, right? But not your small intestine. Okay. Infection, remember, 18 hours plus. So These questions are easy. If you remember the rule and you're reading the stem, if it's less than 6 hours, it's got to be what? An intoxication, okay? If it's over 18, it's got to be a infection, okay? All right. All right. What's pasteurization? Right? You see it, apple juices, all those fruit juices, I see pasteurized, milk's pasteurized. What's pasteurization mean? This is back from chapter 5.1 under definitions, right? Which I asked people to take a look at those definitions. Again, out of 5.1. I sent an email out today too. Hopefully all of you got it. Okay. Remember, pasteurization just means what? We take a, usually add flash heat, okay, for 10, 15 seconds to about 160, 170. And the idea is to kill off usually a main pathogen within that particular food product. Let's say milk, for example. And then it does two things, kills off the main pathogen, and it also then extends the shelf life. Okay. I asked you 5 weeks ago, okay? You were supposed to grab some milk, like a quart, put it in your fridge and leave it unopened for the rest of the semester. Remember that assignment? Because what were we going to do at the end of the semester? We're going to bring it to class, open it and drink it, right? How's it going to taste? Delish, right? Okay? But it's been pasteurized, right? Because what did it do? It just decreased the load of lactobacillus, but there was still bacteria in there. That's why milk will expire in that sense. You can still drink it, just may not taste the greatest for you. All right, so pasteurization does not mean it's sterile, okay? It's just to be killed off. So what's the pathogen we're trying to kill off, like in the milk example? Right? Mycobacterium bovis. Mycobacterium tuberculosis is spread human to human, good old TB, which we'll cover in the respiratory unit, okay? Mycobacterium bovis used to be a very common cause of tuberculosis, gastrointestinal TB in this country. Up until the early 1900s, all right, when they realized is that this bovis was on the udders of cow, this bug was, it got into milk. People drank the milk and they got gastrointestinal tuberculosis. Then they realized is if we pasteurize the milk, we heat it up, we kill off mycobacterium and it extends the shelf life. Okay. All right. So pasteurization. What's pickling all about? What compound are we going to use to pickle something? Right? Salt, right? Usually, right? You're pickling something, you add a bunch of salt. We just talked about mannitol salt agar. So again, salt's a great, right? Great preservative. Section 5.6 was on salt and sugar. Both of them do what? They decrease the amount of available water so bugs can't grow. And that's how pickling works, right? You add a ton of salt, only a few organisms are going to be able to grow in that high salt environment, one of which is staph. Okay. All right. Okay, that's good enough. So on the second section of chapter 30, you can read it over Thanksgiving break. It's FYI, right? It's not on the test, right? It's really about really sort of how many of these products are used and made. All right. Okay, but really what I talk about today here, we got a few minutes for our quiz, right? Just again is here's fermented foods where the foods are intentionally altered, okay, versus fermentation, right? From our definition for metabolism. Okay. Good. All right. Now, right, on food spoilage, it's about, perceived, okay? What do I mean by that? Well, depending on your culture, right, something you may find right, unpalatable in another culture, right, is the norm, right? Same food, just different stages, right? And you may think of it as spoiled, and yet maybe they think of it as, right, more culture. So it's all about, right, perceived tastes. Again, the example I used was yogurt. There's a wide range of yogurts out there, right? Between this really sort of stronger sour ones that you may not like versus the really sweet ones. Okay. Now, why is it you don't want to eat spoiled food? Right? Because generally, what happens is, right? It's not that they look bad or they taste bad, even, right? It's just what happens statistically is as the food starts to decompose, your risk of running into a pathogen increases, okay? But other than that, you could eat a lot of spoiled food. You could eat everything in front of you right here and you'd probably be okay, right? Now, it may not be your top choice, right? But you'd make it okay. All right. All right, so 31.1 or 30.1 now, right, talks about intrinsic versus extrinsic. All right, so intrinsic factors certainly are inherent to the actual food, So intrinsic factors, number one here, tying in again with section 5.6 is this water availability called the AW, is the coefficient, AW coefficient. So if you looked at the videos, we talked about this, right, on water coefficient, right? We talked about salt and sugar, okay? And so, right, and as bacteria, most of them are going to have, of course, the AW of water is 1. So what bacteria had a AW less than 1 that allowed it to survive in sugar environments, high salt environments? It's what poisoned, right? Both of you. Right? Staph, right? Staphylococcus, right? It does well in a high salt environment because it needs less water. Technically, it's AW 0.86. You don't have to know the number. What you need to remember is staph needs less water, okay? So they're going to be selected out, all right? Fungi need less water, okay? So at the end of this semester, right, look in the back of your fridge, the stuff that you forgot about for 4 months. What are you going to find? Mold, fungi, okay? Because they'll be able to grow there, right? And they're going to need less water. Okay, other intrinsic factors, pH, right? Nutrients, do they have a biological barrier, right? So eggs have pretty good protection with their shell, but you can still find bacteria inside them too. So just take a look through those. We got a couple more minutes here and we'll wrap. All right. So, hey, there's our favorite table from section 4.2. Remember, so why is this table so nice? There's your 5 different temperatures, your 5 terms you have to know, people, related to oxygen, okay? Right? A couple of terms on pH, pretty easy, neutrophile, acidophile, alkalophile, and then here we are with halotolerant, right, and halophile, right? The difference being, of course, halotolerant, like staph, means they can tolerate a lot of staph, where halophile means they have to have a high concentration of salt to make it. All right. So, all right. Anyway, there's a couple of slides here just on intrinsic factors, again, about water availability. So again, if you think about it, have you ever had cereals go bad on you? Cereal. They make it stale, but you don't see any fungus on them, right? What type of cereal are you eating? Don't tell me Frosted Flakes.
No.
Fruit Loops. Sugar Pops. Nothing like that. Fiber One.
No.
Oh.
I eat like.
Cinnamon Toast Crunch. Okay, right? So how come that cereal's not loaded? How long has it been in your cabinet?
I don't eat cereal.
Okay, all right, right? But you don't find cereal with mold all over, right? Because there's no water, okay? All right, so again, all right, it takes breads a while, right? Nuts too. So, all right, okay. So here's our buddy staph. So staph and fungi, right, require less water. So they're gonna have an advantage that way. Okay. The example we used in class again with jellies or jams had to do with sugar, right? Remember, there's lots of sugars in them. Those sugars, of course, make water unavailable, so the bacteria can't grow, okay? Because they need water just like we do. All right, okay. All right, pH becomes important when you start canning foods because for acidic foods, as long as the pH is below 4.5, which is pretty acidic, spores can't germinate. Spores like Clostridium, okay, right, or Bacillus, right? So pH becomes important to prevent, right, spore germination. All right, that's all that stuff. Good enough. Okay. All right. So for extrinsic factors, storage temp, all right, things like that. So just real quick, so we'll stop in a minute here for time. How about freezing? That was out of section 562. If you freeze something, how much of the bacteria are killed? All, none, a few? What do you think? What was the stat from section 5.6 on freezing? What do you think it was? Well, it kills half is what it is, okay? But the rest killed half the bacteria. But when you defrost it, the other half kick back into gear, okay? How does freezing kill the bacteria that half it kills? They get crystals inside their cell and then they rupture the bacterial cell, right? But freezing is good for about half of the bacteria, okay? That's about it. But still, I mean, it's still good, right? But just never forget, right? Is that chicken, right, that you froze, when you leave it out on the counter, you don't want to leave it too long, right, before you cook it because the bacterial numbers will crank through the ceiling because of exponential growth. Okay. So the worst thing you can do is pull that chicken out, let it sit there for all day, Then that night decide, you know what? I'm going to Mickey D's instead. Okay. And you put the chicken back in the freezer. Okay. Then you pull it out later, let it set again all day, right? The bacterial load would be very high. Okay. Good here. All right, again, remember psychrotrophs I have bolded. Those are the crew right there that are really going to be the bugs that grow well in your fridge. Again, they like around 5 degrees centigrade, right in that range. Remember, if your fridge is 41 degrees Fahrenheit, it's too warm, people. Bacteria are going wild, right? Fridges should be down about 38, man, 39 max. Okay. All right. Good enough here. That's good. Okay. All right. So we'll stop here. So we got time for our quiz. Today's quiz is on paper. Okay. So in a minute here, Wyatt will be back in a second. We won't keep one for another minute. But anyway, so we won't need your computers, but when we get ready, we'll clear your desk, right? Phones off, smartwatches off, earbuds out, all that good stuff. All right. But it's a 2-page or front and back quiz, about a dozen questions. Okay. Okay.